During an animal disease emergency, diagnostic sampling will be needed to determine the disease as well as identify infected animals. This just-in-time training presentation will overview basic points to be aware of when collecting and packaging diagnostic samples as well as shipping requirements. The diagnostic sample type and collection method needed for an animal disease emergency will depend on the disease of concern, the species of animal involved, and the diagnostic tests to be performed. Regardless of the diagnostic sample to be collected, a few steps should be followed before you begin any sampling procedure. First, check with the diagnostic laboratory or your team leader to determine the appropriate samples to collect as well as how they should be handled, including any selective or transport media or temperature requirements. Gather the necessary supplies you will need for sample collection as well as any personal protective equipment needed. In the event of a highly contagious or foreign animal disease, strict biosecurity measures will be essential and will therefore require personal protective equipment such as gloves, coveralls, and footwear that can be disinfected or disposed of following the sample collection process. In the case of zoonotic diseases, additional PPE such as respirators or face shields may be needed. If possible, pre-label specimen containers to ensure all required samples are collected. All samples must be labeled clearly and legibly with pencil or waterproof ink. The label should contain sufficient information to allow traceback of the sample to the premises and individual animal or pen from which the sample was taken. When a variety of sample types are taken, such as swabs from multiple locations or tissue samples from more than one organ, the label should clearly identify the origin of the sample. The date the sample was taken should also be included on the label. A sheet may be included with a group of samples from a single premises to aid in matching the samples with the animal, pen, or group number, and to add additional information such as sex or age of the animal. Samples to collect may include whole blood or serum, culture swabs, skin samples, or tissue samples. If whole blood or serum samples are required, ensure the correct blood collection tube is used. Blood tubes contain different types of additives which can affect the diagnostic tests. Most blood tubes can be distinguished by the color of the tube top. Swabs of the mouth, nose, or rectum may be requested for some diseases. Individually packaged swabs with culture media or sterile swabs with or without preservatives may be provided for diagnostics. With either type, be sure to avoid touching the swab tip to anything other than the sample area being swabbed, and place the swab back into the sterile vial immediately after collection. Skin samples such as skin scrapings, ear punches, or ear notches may be needed and should be packaged as instructed by the diagnostic laboratory. Lastly, tissue samples collected during necropsy may be requested. These samples may need to be submitted in a preservative such as formalin, refrigerated, frozen, or a combination of methods. Each sample should come from an individual animal, not a pool of animals unless specified by the receiving diagnostic laboratory. Samples must be collected and handled in a manner to prevent degradation and maintain the integrity of the sample. This may involve placing the sample in selective or transport media or keeping it chilled. Samples preserved with formalin must be in a sample jar that contains at least 10 parts liquid to 1 part tissue. Handling of tissues and fluids from a suspected premises or animal will require strict adherence to biosecurity and infection control procedures. Wear disposable gloves and change them frequently. Use disposable equipment such as needles, syringes, and blades whenever possible, and change equipment between each animal. These practices serve not only to prevent sample contamination, but to prevent the accidental spread of disease between animals. When non-disposable equipment must be used, such as necropsy tools or surgical instruments, clean and disinfect equipment between each tissue and each animal. All equipment used to collect and transport samples while on the premises should be cleaned and disinfected in the designated area prior to leaving the farm. Care should be taken not to contaminate diagnostic samples with disinfectant, thereby inactivating it and providing false negative results. After diagnostic samples are collected, they must be prepared for shipment to the appropriate diagnostic laboratory. The necessary sample packaging will be dependent on the agent suspected and the type of samples being sent. We will review basic points to remember when packaging samples. However, additional measures may be required depending on the situation. It is essential to assess and ensure that packages will be able to withstand shocks and vibrations during shipping, pressure changes, and other conditions encountered during transport, such as weather, temperature, and rough handling. Therefore, when packaging the diagnostic samples, it is important to do this in a manner that will prevent breakage as well as spills and leaks, maintain the appropriate temperature requirements, and have the necessary shipping labeling. Fragile primary sample packaging, such as tubes, slides, and vials, must be wrapped and separated to prevent breakage. In the event the sample does break during shipment, care must be taken to prevent samples from spilling or leaking outside the container. This is accomplished by using 2 waterproof layers of absorbent material, such as paper towels, between them. Preventing spills or leaks is particularly important given the potentially infectious nature of some samples during an animal disease emergency. In order to be of diagnostic value, samples must also be protected from extreme heat or cold, and some samples require refrigeration or freezing. Finally, the package must be labeled appropriately to alert shippers to the nature of its contents and to provide contact information for the diagnostic laboratory. When possible, use plastic screw cap containers and avoid glass containers. In order to prevent breakage and spilling of samples during shipment, it is important to pad them well and include spill barriers and absorbent materials in the packaging. Primary sample containers such as blood tubes should be well padded with material such as bubble wrap, particularly if multiple containers will be shipped together. This should be placed within a primary waterproof barrier such as a canister or plastic bag, which contains material such as paper towel sufficient to absorb the samples should it spill. This should be placed within a second watertight barrier and finally into a rigid outer shipping container such as a cardboard box. Formalin fixed samples should not be packaged in the same box as non-formalin fixed samples. The left photo shows several sample tubes packed in absorbent padding material to prevent breakage and spillage during shipment. These will then be placed in the white plastic container on the left for leak prevention. The right photo shows a shipping box with dividers to keep samples secure during shipping. In order for samples to arrive at their destination with diagnostic value, they must be maintained within the temperature range appropriate for the sample type. Samples shipped on ice should have a layer such as bubble wrap between the sample and the ice packs to prevent freezing of the sample. Follow any requirements specified by the diagnostic laboratory. Swabs for PCR and tissues preserved in formalin can be shipped at ambient temperatures unless stated otherwise, but should be still protected from excessively hot or cold conditions. For samples that require shipment on ice or dry ice, it is crucial to maintain the necessary temperature, referred to as the cold chain, from the time of collection until arrival at the diagnostic lab. Otherwise, such samples may lose their diagnostic value. This photo shows a sample for submission in a sealed plastic bag to prevent leakage. A cold pack has been placed with the sample to keep it chilled during shipment. When packaging samples that require ice for shipment, use only ice packs, picnic packs, or sealed ice containers. Never ship with cubed or crushed ice, even if it is sealed in a plastic bag. Samples should be protected from direct contact with ice packs to avoid being damaged by freezing temperatures. Samples shipped on ice should arrive at the diagnostic lab within 24 hours. Packages shipped with dry ice are considered dangerous goods and may not be accepted by some carriers. It is essential to take measures to prevent leakage during the shipment. Line the box with a plastic bag in which to further enclose the sample contents. A dry ice label must be affixed to the outside of the shipping container, and such shipments are considered Category A shipments, which will be explained later. Dry ice may inactivate some viral samples, so be sure to seal the sample vials or test tubes tightly if shipping with dry ice. The sample submission form should be sealed in a separate plastic bag, and non-formalin samples should be shipped in a separate box. Samples collected during an animal disease emergency will need to be sent to the USDA National Veterinary Services Laboratory for diagnostics. Special shipping requirements exist for infectious disease samples. The packaging and labeling of biological substances for shipment requires familiarity and training with current rules and regulations, which frequently change. Shippers are responsible for proper packaging, marking, labeling, documentation, classification, and identification of each shipment. The transport of biological materials is regulated by the Department of Transportation Hazardous Materials Regulation, Section 49 of the Code of Federal Regulations. These regulations impact the packaging and labeling parameters of the samples being shipped to the diagnostic laboratory. The Department of Transportation defines a hazardous material as any material that is capable of causing risk to health, safety, and property during transportation. This definition includes infectious substances and animal diagnostic samples. Samples collected during an animal disease emergency are considered infectious substances as they are known to contain or reasonably expected to contain pathogens. The United Nations and the United States Department of Transportation specify how these potentially biohazardous materials should be classified and labeled for shipment. Category A infectious substances are those considered capable of causing permanent disability, life-threatening or fatal disease in humans or animals when exposure to them occurs. Category A substances are further divided into those that affect animals only, which must be labeled with the code UN 2900, and those that can affect humans, which should be labeled UN 2814. Pathogens of concern in an animal health emergency can be of either type. Category A infectious substances require specially rated triple packaging materials and must include a declaration of dangerous goods form on the outside of the box. Category B materials are those not in a form that can cause permanent disability or life-threatening disease in otherwise healthy humans or animals on exposure. Category B materials include specimens for general, diagnostic, or investigational purposes. Must have leak-proof triple packaging and should be labeled on the outside of the box with the code UN 3373. These designations impact the packaging, labeling, and shipping parameters of the sample, which must comply with federal regulation. These photos show UN package certification markings required for shipping all infectious substances, Class 6.2. The UN identification number in the upper right-hand corner of the right photo is required to properly identify the type of sample included in the box. This box is labeled UN 2900, containing a Category A infectious substance, animal only. Packaging for Category B infectious substance includes placing the solid or liquid specimen in a leak-proof primary container and labeling the content. Place the primary container inside a flexible, sealable plastic bag or rigid secondary container. Place absorbing material inside of the secondary container. Place the secondary container in an improved, insulated, waterproof, flexible or rigid shipping container. Place ice packs inside the shipping container. Place case histories and submission forms in a sealed plastic envelope inside the shipping container. Seal the shipping container. Ensure the appropriate package marks are on the outside of the shipping container, and then ship. Packaging for Category A infectious substance will require additional precautions and labeling, including an itemized list of contents inside the package and an infectious substance label and proper UN number on the outside of the shipping package. For primary and secondary packaging for Category B includes leak-proof receptacles. Category A requires watertight packaging. The category designation also determines the quantity of sample that can be shipped within a single container. This will also be dependent on the mode of transportation. Materials that are infectious to humans or animals cannot exceed 50 milliliters or 50 milligrams when being shipped via passenger aircraft or rail. However, up to 4 liters or 4 kilograms can be shipped on a cargo aircraft. For Category B, biological substances, up to 4 liters or 4 kilograms of biological substances can be shipped in passenger or cargo aircraft and by rail. When shipping to federal diagnostic laboratories, the type of agent will determine where the samples are to be sent to. The addresses of both National Veterinary Services laboratories are listed. The specific routing instructions for a foreign animal disease diagnostic sample to these labs, including which lab submissions should go to which lab, can be found on the USDA APHIS website. Testing for some high-consequence diseases may be allowed at select APHIS-approved laboratories in the National Animal Health Laboratory Network. A current roster of laboratories in this network and the testing that they are approved to perform can also be found on the USDA APHIS website. Samples being sent to the USDA National Veterinary Services Laboratories will require a diagnostic sample prioritization for processing. This is based on the disease suspected. Priority 1 is used when prompt laboratory diagnostic information is required and the sample is highly likely a foreign animal disease or emerging disease incident specimen. Once received, these samples will be unpacked and examined immediately upon arrival. The NVSL must be contacted by phone prior to shipping a Priority 1 submission. Priority 2 samples are of intermediate suspicion and obtained from situations where a foreign animal disease or emerging disease incident is possible. Samples will be processed the day they are received. Priority 3 samples are those that are unlikely to be a foreign animal disease or emerging disease incident and cannot be distinguished from an enzootic disease or condition. These samples will be processed in ascension order unless otherwise directed by the laboratory director. Priority A designation involves situations where animals in commerce are being held pending the result of the diagnostic testing for a foreign animal disease or emerging disease incident, or other situations where rapid laboratory tests are needed. These submissions will be processed immediately upon arrival and require contact with NVSL before submission. When choosing a carrier to ship biological substances, there are a few important aspects to consider. First, choose a carrier that will transport potential hazardous infectious substances. In accordance with the DOT's hazardous materials regulations, carriers must have security plans when shipping certain agents. Some carriers may not have extensive experience with transporting potential dangerous biological samples, so a full assessment of safety features is needed. Next, choose a carrier that allows tracking so that progress and location can be monitored. Prompt and reliable delivery is key for reasons of security and sample viability. This presentation provided a brief overview of sample collection, packaging, and shipping information. Additional details on this topic can be found on the USDA APHIS National Veterinary Services Laboratory and the Department of Transportation Hazardous Materials Regulations websites. Development of this presentation was by the Center for Food Security and Public Health at Iowa State University through funding from the Multi-State Partnership for Security in Agriculture.